Good afternoon. Thank you, Steve, for that warm introduction. It's my pleasure. I'm lucky to be here, and I'm really excited to tell you about 3 projects, one of which is a longitudinal research project that I think is finally in the home stretch, and then 2 other sort of assay development projects, one of which is also, I think, getting close to wrapping up, and the other is really just beginning. So I want to start with a case that has stuck with me since residency and really is why this long research project blossomed. So this was a 67-year-old guy with relapsed CLL. He was 3 weeks out from induction chemotherapy in anticipation of CAR T-cell therapy when he presented with a fever and headaches, altered mental status, and some falls. And of course, in this patient population, who really don't have any neutrophils at this point, which you can see at admission, there we go. Very low neutrophil count. We're worried about an invasive fungal sinusitis. And of course, by now you figured out that's exactly what this was. So you can see there are abundant septate, non-pigmented hyphae in this tissue. There's some necrosis in the background, some fibrin in blood, and then probably most of his neutrophils are up here at this site of infection. And at surgery, I think a lot of things went right. So he had surgery almost right away. Hyphae were seen at frozen section. They were reported. There were no invasive hyphae identified. On the other hand, we sort of assumed this was an invasive infection. And from the microbiologic perspective, a lot went right as well. Multiple cultures were set up. Multiple PCRs on fresh tissue were set up, but they were all negative. And so, unfortunately, the patient does not do particularly well. He has an ischemic stroke. He goes to the ICU. The asterisk indicates where we finally did see invasive hyphae. At this point, they're in the CNS vasculature in the cavernous sinus. You can sort of see these spikes in his neutrophil counts or where he's getting granulocyte transfusions. And along the way, he's on amphotericin, eventually posaconazole, and months later, when we eventually, sort of in a passing conversation with one of the ID fellows who happened to also be taking care of this patient and was down in the AP resident area to talk about a different case, it prompted a broad-range fungal PCR on this, and we identified an Aspergillus fumigatus. He was transitioned to voriconazole and was able to go to a skilled nursing facility, still with residual deficits from his stroke. And so I don't know that if we had identified this organism up here and transitioned him to voriconazole, he actually would have had a better outcome. But the reason I think it might be possible, and the reason that this sort of stuck in my brain and haunts me a little bit, is because there's a seminal paper in the early aughts that identified a significant survival benefit for patients with invasive aspergillosis who are started on voriconazole and not amphotericin. So this was a randomized controlled clinical trial across multiple sites, multiple hospitals in multiple countries, almost 300 patients, and there was a really significant survival benefit. 53% of people who were started on voriconazole were alive at 12 weeks, as opposed to only 31% for those who received amphotericin. Still a pretty dismal survival with this disease. Now, the idea that you should treat aspergillus with voriconazole up front stands in stark contrast to invasive mucormycoses, where amphotericin is the mainstay of therapy. And in another seminal paper from the mid-aughts out of MD Anderson, the authors identified that if by 6 days after your onset of symptoms, you start amphotericin, your mortality is about half compared to if you start later. So at 4 weeks, it's about 35% mortality, about 49% at 12 weeks, and that's in the appropriately treated group where amphotericin started early. It gets up to the 80% range at 12 weeks if you don't start it in time. And so this is why being able to identify what fungus you're dealing with in cases of invasive fungal disease is particularly important. Before we talk about the identification steps, I want to just look a little bit at the patient population. So the initial patient I was talking about had very low neutrophils, but how low does your neutrophil count really have to be? So this is a recent study that came out of our colleagues here in ENT, Greg Davis and Ian Humphreys, as well as Rod Schmidt, mostly retired from pathology. And there are just a couple of key points to which I want to draw your attention. The first is that a third of their patients had absolute neutrophil counts greater than 1,000 cells per microliters. Normally, we think of the patients being at risk as having less than 500. Interestingly, having a higher neutrophil count really didn't correlate with improved survival. you can sort of get a sense that maybe at 3 months in this high ANC count group, there was a little bit of a survival benefit, but it really didn't cross into statistical significance. There were a couple of factors that did predict outcomes. So having what these authors termed atypical molds, Fusarium, Scedosporium, Alternaria, for example, was correlated with a poor outcome, and getting surgery was correlated with an improved outcome. So these are both surgical and medical diseases. So in terms of identifying fungus, the reason we want to have a sort of broad range organism agnostic approach, culture or PCR, is because trying to do this based off of histopathology is highly error prone. There's now a well-developed body of literature around this, including a couple of important papers. This one out of Stanford by Sangoyan is one of the better, one of the more well-known, and one of the important findings is people tend to, the pathology lore is that one of the reasons you can misidentify things is, patients may have gotten antifungal therapy and this may change the morphology, and actually that's probably not the reason we misidentify them. The reason is they just look a lot alike. And so there are evidence-based guidelines that have been developed. Our colleague Jeanette Guarner at Emory and Mary Brandt have published these in a wonderful review. And so we try and break down the molds into septate hyaline molds, which includes Aspergillus and others. The pigmented molds are the Dematiaceous molds, and that includes organisms like Alternaria, but frankly, you often don't see the pigments. And so these two groups sort of, there's a fair amount of crossover. And then finally, they're the pauciseptate molds, which really don't have septations except at branch points. And those are the mucorales of the zygomycoses. So we try and put these into groups, into broad categories, but it still matters even within the septate molds. If you can get an ID, yeast you try and sort of break down by small, medium, and large and give her differential. So what if you say, well, Josh, I'm going to play the odds, right? Aspergillus is one of the more common organisms. Why can't I just say this is a septate hyaline mold? So you should probably be thinking about Aspergillus. And the reason is, this is data that we collected from the paranasal sinusites. That's a mix of reference lab work and internal patients for PCR and internal patients for culture. This is from 2011 through mid-2016. Aspergillus doesn't even constitute a plurality of cases, including multiple species. And so a fume ranges between about 12 and 23%, depending upon which assay you're looking at. But if you include all Aspergilli, you're probably only getting up to about 35% of cases. There are two other important features I want to point out. One is that sometimes we just can't get an ID by culture. I think this is less common now that there's fairly close collaboration between the mycology lab and the molecular lab. So we do a fair amount of identification by sequencing. The second, not shown here, is that about 12% of these cases are polyfungal. So the other big pitfall, of course, is that you may see something that suggests one organism and completely miss the other that's hiding in the background. And it's very humbling when that happens. I think I've made that mistake, and I know many of my colleagues have as well. And it's hard because there's a patient on the other end, and you're really just trying to get them the best care. So sort of putting all this together, I developed a number of questions that just wouldn't leave me alone about how we can use PCR to really bridge what happens in the clinical lab. And as I showed you in that first case, sometimes we don't have an answer for our colleagues on the clinical side of things. And how do we bridge that with what's in the histopathology lab, where often we have diagnostic material? And so, some important questions that I was hoping to answer were, what are the advantages or drawbacks of PCR? And can we really see if this makes a difference in patient care? And finally, I think this is a really important question: whom do we test and when? And so I'll try and answer some of those as we go forward through this first part of the talk. First, let's talk a little bit about what's known. So this was work that came out of Stanford at the end of 2017. They have a very similar assay to our broad-range PCR assay, and there are a couple of points to make from this. They sort of had a two-phase study. First, they took 60 known positive cases where they had an organism ID'd by a reference method, 57 cases that were negative by reference methods, and they used that to test sensitivity and specificity of their assay. They followed that up with 116 culture-negative patients, all of whom had suspected invasive fungal disease. So you can see high sensitivity and specificity. The true specificity is probably higher because one of their negative cases was actually, they believe, the PCR result clinically. And of their positive cases in this phase 2 part of the study, almost 60% of them, 60% of those PCR results led to a change in management, mostly in medication. In a couple of cases, it changed surgical management as well. So there are two other key points that I take from this study. One is that the submission type has an important impact on diagnostic yield. And so what they found is that if you had a sort of open surgical resection where presumably you had bigger tissue, you had a much higher diagnostic yield, somewhere in the 70% range. Smaller specimens like core needle biopsies were somewhere around 50%, and they really didn't see any yield from fine needle aspirates. The final point that I want to make about this study is when they compared the diagnostic yield from FFPE tissue, formalin-fixed, paraffin-embedded tissue from the histopathology lab, from their anatomic pathology colleagues, performance was about the same as for fresh tissue. And so we'll come back to that and try and explain why. It's become sort of popular to refer to some of these broad-range fungal assays as universal PCR. I think in general, when I see this in the literature, this means at least two of usually University of Washington's broad-range PCRs were ordered on a specimen. That's a mix or all three of the broad-range mycobacterial, fungal, or bacterial PCRs, often with next-gen sequencing as a reflex. And so this was a nice small study, but with really detailed chart review, in which for all of their samples, they identified a diagnostic yield of about 30%. And in about a quarter of all of those samples, they made a significant clinical change. Now, that included both positive and negative results, which I think not looking at negative results is a shortcoming of some of these studies, but can be quite informative. The other thing that I'll point out is that having corresponding sort of indicia of inflammation, or ideally organisms seen by histopathology, increases your diagnostic yield, right? This is the gospel of pretest probability. I don't think that's a surprise, but I do think it's important when you're thinking about what specimens to select. And I think it also goes to the notion that why FFPE specimens probably perform about as well as fresh tissue. In this study, they used sort of a clinical judgment as their gold standard for whether patients really had an infection or not, and they estimated sensitivity of our assays, this group of 3 PCR assays at about 51%, specificity at 94%, and you see the positive and negative predictive values. So I want to look at one more study before we get into our data, because there are a couple of key points. This has just been accepted in the Journal of Clinical Infectious Diseases. And again, same sort of situation where universal PCR is some combination of our 3 primary broad-range assays. And what I really take from this figure is that anatomic site makes a difference. I think this is probably more to do with what's most likely to be infected as opposed to anything intrinsic to the tissue. So this is concordant with our anecdotal sort of sense in our lab, but heart valves tend to be pretty high, have high diagnostic yield, also pleural fluid. Interestingly, in this data set, sinonasal specimens only had about a 10 to 12% diagnostic yield, but most of those results changed management. So the study only looked at positive results, and of those positive results, about half changed management. And like in the previous studies, the presence of an organism or inflammation on some sort of concurrent study increased the probability of diagnostic yield, although they didn't quantify that, and they found that the yield from FFPE was similar to fresh tissue. They didn't look at volume of specimen in the same way that the Stanford study did, but they thought that tissue being submitted, in which category they lumped FNAs, was more likely to have a diagnostic yield than fluid. Again, that may speak more to selection of tissue for submission rather than anything else. So I want to turn our attention to a study that I started a number of years ago as a resident and really has been completed by Alex Mays, who's built a really wonderful set of informatics tools along with Patrick and Nathan Bright that I'll tell you about as we go along. But we pulled data from the LIS from 2011 through 2019 inclusive which represent for all fungal testing that we've done. It includes about 38,000 broad-range fungal PCRs, 28,000 fungal cultures, representing nearly 50,000 distinct patients. We excluded the vast majority of these because they are not from the paranasal sinusites, and that was really, we were trying to limit our focus. And then when we just look at sinonasal specimens, we see there are about 640 fungal PCRs representing nearly 600 patients, 427 cultures. Really all of those come from the inpatient setting here at UW or one of our associated institutions. So for our inpatients, there are only about 55 patients, which is a tractable number for chart review, which is great. And then they had about 78 fungal PCRs between them. So let's try and march through this and see how our assay performs in a sort of a high probability of infection setting. So first, what we notice is that the positivity rate is higher for broad-range fungal PCR. I was actually surprised that it was almost 50%. It came in just at about 49%. Culture also came in higher than some preliminary data I had collected at right around 20%. And so we asked the question, is this reflective of all tests, or is this really something about our patient population? And so the way we went about that was to say, all right, here on the left, so this graph is all patients with samplings from the paranasal sinuses, and so that includes our reference lab clients. For all broad-range fungal PCRs, we're at about 10%, which is consistent with some of the previous literature I cited. But in the sinuses, even when you look at our reference lab clients, we're up around 36%. And so the way I make sense of this, particularly now that I've had a chance to sign out a bunch of these cases and review the pathology reports that come in with them, there are a lot of specimens, particularly in this non-sinus, this is all sites, but in the non-sinus category that come in, and there's granulomatous inflammation. It's a nice, dense epithelioid granuloma, and the suspicion is sarcoid. And the clinicians would like to do broad-range fungal, mycobacterial, and PCR testing to rule out an infection so they can put the patient on steroids and treat their sarcoid. And so that emphasizes that while I can't quantify it yet, these negative results are actually still oftentimes quite informative, and also explains why the sinus population, I think, is a really high positivity rate. I don't think of sarcoid as being particularly prominent in the sinuses, but I do think if you have a reason to order a broad-range fungal PCR on a sinus specimen, you're probably thinking about someone who's quite sick and may have an invasive infection there. So we divvied this up a little bit differently because we did have some outpatients listed. In the outpatients, so you see the counts here on the left and the proportion positive more clearly on the right. So obviously the bulk of our patients are from outside clients. We have a small population from here. And then we have a few outpatients who actually their PCRs are not positive as frequently as in the inpatients. And again, I think this just goes back to who's sick. And presumably, if you're getting sampled as an outpatient, you may be suspicious for an invasive fungal infection. But you may also be sampled for an allergic process. And so we're probably including people who don't have disease in here, but where we want to rule things out. As opposed to the inpatients, we're probably actually quite worried. So I want to turn our attention to this question about formalin fixation, because I think it's a really important question. We've certainly done experiments in the lab where we've seen that prolonged formalin fixation time destroys DNA. This is, I think, a known phenomenon, right? It's a complex biochemical process. It leads to DNA cross-linking and shearing and reduces PCR yield. And so we tried to break this out first by count and then by proportion, so same data displayed in two different ways. And what you see is that actually it's about the same. This difference in percent positive is not statistically different. It's about 42% positive in the FFPE samples. It's only about 35% in the fresh specimens. And so I think the question is, how do you overcome the damage caused by formalin fixation or other pre-analytical variables? And so I think if you're a pathologist or if you're working with a pathologist, you get to say, oh, look, block A1 has not many organisms, block A3 has a bunch of organisms, but it also has bone and went through a strong acid decalcification, which is much worse for DNA, I would argue, than formalin. And so I'm going to pick block A2, which is plenty of organisms. So I really think that's one of the key links between the molecular assays, the clinical lab, and the anatomic pathology side. So one of the hard questions to answer with this process is, who wins the race? Is it PCR or is it culture? And so this is all, the big caveat here is this is all final result dates. And I think we're all pretty accustomed to having preliminary results come out well before the final dates. So we see the negatives and positives in red and teal respectively for the broad range fungal PCR and our fungal cultures. And we're several days faster by final result on the PCR. There are a couple of other complexities here. One is I haven't tried to take into account how, I mentioned how the prelims come early. In the case of the fungal PCR, it's probably anywhere from 6 to 36 hours earlier. I think that range is much broader in culture, where sometimes rapid growing molds we can give a presumptive diagnosis quite early. I also think there are probably some outliers that are positive out here, which may be because we're waiting to final them until we have sensitivity data back, but I have to dive into some of those cases a little bit more. And then the other sort of big elephant in the room that I haven't touched is, how do other assays that we run in the molecular microlab that we often set up front with broad range PCR, like our Aspergillus fumigatus specific PCR or a Zygomycete group specific PCR, how do they factor into this? Because we result those, or they're able to be finaled about 2 days faster than the broad range fungal PCR. They don't have the same breadth of organisms that they can detect, but for the organisms that they do detect, they're more sensitive. And so in terms of how to piece all these together into a better model of how we actually should be algorithmically testing patients, I don't have a great answer yet. One of the things I do want to point out about turnaround time is, while turnaround time is pretty constant for culture over time, we've gotten faster in the molecular microlab. And so it'll be interesting to see if this drop in about 2.5 days turnaround time continues, as we've now, Tatjana Vlasic and Dan Hoogstraat have built a really phenomenal informatics sort of architecture that allows us to track, manage, and sign out our cases. And my suspicion is that over time, this is going to shave off a little bit more time from our results. So for those 50-odd patients who are UW inpatients, I was able to dive in and do a chart review and tease out a couple of, I think, really useful findings. The first is that PCR led to a medication change, positive PCR results led to a medication change about 18 or 19% of the time. PCR was the only positive diagnostic test in about 9% of cases. And so if I understand how to do this correctly, that means the number needed to test is about 11. I think that's a fairly conservative estimate, but a reasonable first order approximation. Come back to that on the next slide. There are a couple of other points I want to make, which is that negative testing is still valuable. So there were a number of cases, about 12% of all the tests ordered, where the clinicians were more comfortable not adding antifungal medications because the testing was negative. Some of that includes culture results, but one of the big advantages you have from the broad-range fungal PCR is we result it potentially weeks faster than a negative fungal culture. So there were 2 cases that were deemed to be colonizers or bystanders, and this is really an issue of clinical judgment. In one case, they were able to de-escalate, in the other, they did not add additional therapy. And I think it was really striking because you read the notes and it's pretty clear that this patient is not immunocompromised. They've been using Afrin a lot, and so that probably explains the local necrosis in the nose. And so then you have sort of superinfection or colonization by some of these potential pathogens, and you just have to watch the patient clinically. The other thing I'll point out is that this is still not good for patients, right? The 30-day mortality, just looking at these cases, was about 14%. So I want to talk a little bit about that number needed to test, because one of the questions is, how much does it cost, right? And obviously, I hate to boil down a patient's life to how expensive is it to treat you, but I think in a day when resources are constrained, it's important to at least think about. And so the cost of 11 fungal PCRs is about $5,000. In 2019 or 2020 dollars. In 2017 dollars, the cost of 1 day of a regular hospital patient admission in Washington State, on average, hospital expenses, is about $3,500. that trend has not, the rate of increase has not abated. And it strikes me that shaving off one regular inpatient day is about the minimum impact you can have. And so I can't yet attribute sort of changes in length of stay to whether patients got broad-range fungal PCR. But I think that's sort of, if that's the minimum, I think we have real opportunities to, at the very least, break even, to say nothing about getting patients on the appropriate therapy early and potentially improving mortality. You know, to continue to be crassly financial, protect a $200,000 to $800,000 investment in their leukemia care. So I do think there are potential for cost savings, and that it does make financial sense to add these up front, particularly when we look at the ability to catch infections we might otherwise miss. So I'm hoping to answer those questions with help from our fabulous informatics team. So Nathan Bright and I just spoke yesterday about how to make use of this data that he and Patrick were able to pull in an architecture that I hope that seems to dovetail well with the electronic data warehouse, and maybe like the sort of analysis package that Alex Mays built, is reusable for other tests and other anatomic sites. So Nathan pulled a mix of emission, discharge, and transfer data, as well as length of stay, from which we'll derive length of stay data, and sort of a robust look at medication changes. And the goal is to compare our 255 patients who only receive culture to those who only receive PCR. And one can imagine also doing this for just crude aggregate hospital costs to try and see if this single variable makes a difference. So I want to wrap up this part of the talk by really emphasizing that I think broad-range fungal PCR does not replace culture, but is a really important high-yield component, particularly for high-risk patients. And so this may be the only diagnostic result, and working with the pathologist to make sure that you pull tissue from your anatomic pathology lab is a really important way to not miss infections. Just to re-emphasize, medication changes do happen with positive results, but negative results are informative too. So I want to move on to talk about an assay that we have in development where I think we've made some really nice progress. And this is really sort of thinking about what happens when small intracellular pathogens look alike and you're trying to tease them apart, or, as in this first case, when you actually don't see anything. And so this was a case that came through when I was a resident then on CP. And this was a 30-year-old guy who was returning from the Peace Corps in South America. He had a history of latent TB, for which he had claimed he had been treated. He also had an extensive travel history throughout South and Central America and Australasia. No history of cutaneous lesions, no other symptoms, and this mucosal lesion in his nasal cavity had been there for several months by the time he presented to our ENT docs, who were thinking about chronic erosive processes like rhinoscleroma, which is a chronic Klebsiella infection, or mycobacterial infections. And you know, the histopathology is actually not all that informative, although it is striking. There's a rich influx of lymphocytes and histiocytes, which we see here. There's a little bit of necrosis that's sort of patchy, but nothing really diagnostic even after multiple rounds of culture, multiple biopsies, the whole panoply of special stains for microorganisms in the anatomic pathology lab. And so ultimately, an astute ID fellow was examining this patient and palpated an enlarged cervical lymph node, at which point she said, I know he doesn't have cutaneous lesions and claims to have never had them, but he has an erosive mucosal lesion, he's been in an endemic area, and he's got an associated lymphadenopathy. I think this is mucosal Leishmaniasis. And about 3 months after he first presented to us, the CDC came back having detected Leishmania brasiliensis in this patient. So you may be more familiar looking at slides like this, where you can see a mastigotes running around inside of histiocytes, although it's not unreasonable to also consider something like histoplasma in the differential. And in the first case I just showed you, even though we did broad-range fungal PCR, we did not detect Leishmania. In this case, we did. We reported as an incidental finding. The 28S locus that we use is a region of pretty high conserved sequence, fairly conserved sequence. And so that's how we usually detect these organisms. The flip side, though, is that locus is conserved enough that we can't identify the species. And so that's why I'm focusing on mucosal Leishmaniasis So I want to just take a minute to introduce that disease. Mucocutaneous Leishmaniasis is usually thought of part of a spectrum. There are about 1 billion people who live in endemic areas. There have been about 1 million cases over the last 5 years. And mucosal Leishmaniasis can not only be disfiguring, but places patients at risk for superinfection. And so on the right, we see sort of a classic cutaneous Leishmaniasis lesion, which can either progress or present without a cutaneous lesion into some of these erosive, highly destructive lesions, which are not only if nothing else, disfiguring. This is actually not an extreme presentation of mucosal Leishmania. I had the opportunity to see a number of mucosal cases when I worked in Brazil in a strange sojourn we can talk about some other time. But this is not an unreasonable presentation. So there are 2 other pieces of this information, or 2 other things that I want to draw your attention to as I introduce this disease. First is, so there's this sort of mucosal belt across northern South America where mucosal Leishmaniasis is more common. The other thing is you can see it's distributed around the world, and these different colors indicate different species. And I don't want you to pay attention to which species is where necessarily, simply to note that there is significant overlap between their geographic regions. And so that's not enough to tell you what species of Leishmania the patient has. And actually, the species that the patient has matters quite a bit in terms of treatment. There are other factors that are involved in deciding whether or not you're going to treat the patient. How big is the lesion? How many lesions are there? Are they immunocompromised? Do they have visceral disease? I'm not going to talk about Kala-Azar today, but it's highly fatal, so it's always treated with pretty nasty drugs. And then the cutaneous disease, so we can really divide our sort of Leishmania species into either old or new world. In the old world Leishmania, I think the treatment decision is really, is it Leishmania tropica, which is more likely to resolve on its own, although expert opinion diverges somewhat. And then within the new world species, subgenus Viania is the real question. This includes Lesh brasiliensis, guianensis, and panamensis, which are much more likely to produce this mucosal disease. They have this mucosal tropism. So we wanted to develop, ideally, a single locus PCR assay that could distinguish these different species. And so when Kendall Jensen, our molecular pathology fellow, was on rotation with us, he did a really helpful literature review and identified several candidates. The other features of this potential assay would be that you have sort of flanking conserved primer binding sites with an intervening region of high sequence and potentially length polymorphism. And it has to work across a pretty wide range of, from a PCR perspective, fairly hostile conditions, right? So for comparison, E. coli is about 50% GC, human on average is about 41%. The true Leishmania genomes are up in the 70 to 80% range, and the Viania subgenus down around a scant 60%. So really kind of a wide range of molecular conditions to deal with. And so one of the target that we thought had really good phylogenetic discriminatory power was something called the mini exon repeat gene, or the spliced leader gene. And this is both cool biology and a couple of useful factors as we design the assay. So it's, first of all, arrayed in about 100 or 200 tandem repeats, which means there's this sort of antecedent template amplification step. So a single genome has multiple targets. They're highly conserved potential primer binding sites. And what's really cool is this 39 nucleotide highly conserved region gets transcribed and then trans-spliced onto the 5' end of other mRNA species throughout the organism, which is just really kind of cool biology. It doesn't really influence what we're doing, but it is really kind of cool. So we were lucky enough to have one of our talented MLS students, Carissa Crawford, join us in the lab, and in a short month, she was able to take this from concept to really what seems to be a working assay. So you'll see that, and I should point out that many of these strains we either purchased from ATCC and then cultured and extracted DNA from those promastigotes, or we were kind enough to get cryostocks for culture or extracted genomic DNA from 2 of our collaborators in infectious diseases, Wes Van Voorhees and Fred Buckner, who've been really, they and their staff have been really generous with their time and their reagents and their expertise. And so you can see where it falls in the gel that's over here that I'll walk through in just a second, what we estimated the amplicon length to be, and what we expected the amplicon length to be. And so there's a range of sort of intensity of bands, which probably inhibited our sequencing, which I'll talk about in just a second. But one of the things that I really thought was interesting was for this L. donovani species that we bought from ATCC, it's 300 bases when we amplified it out of the genomic DNA, when we amplified the mini exon gene. And that's significantly lower than we would expect. And so at this point, it was about the end of Carissa's rotation. I was getting ready for this talk. And so we were able to get this sequenced in the clinical molecular lab by E.G. McGuire. Was able to find time in between. I'm very impressed, in between reviewing cases for NGS. And I think we have some answers. So the first is that we just picked a one-size-fits-all dilution for these, and so I think we wound up both over and under diluting them, which explains a lot of the poor sequence quality that we see, and some of the misidentifications, but mostly we're able to do a good job identifying. So let's just walk through this briefly. The positive control, we took the mini exon gene from lizard Leishmania, L. tarantulae, and I changed every 5th base, synthesized that in a plasmid from IDT, and sure enough, this was, our positive control was 81% identical to lizard Leishmania, 100% identical to our positive control, so great, that's working well, that was high sequence quality. We had one sort of challenging identification where this L major strain was either L major or Leishmania mexicana. I'm a little bit curious to dig into the strain and just make sure it's accurately annotated. The major error that we had was this L. tropica strain was identified as either L major or L mexicana, but again, low sequence quality. So it'll be interesting to see if we can troubleshoot the sequencing and resolve this. The L. donovani strain that didn't make any sense to us turned out to be Leishmania amazonensis. So we were working with the Lesh amazonensis, and we sort of went through the standard lab medicine resident call, right? When could the specimens have been swapped? Chris and I were very careful doing this. I really don't think that we swapped them. We do have other specimens from other points in the culture, and so we'll do some testing of this. But ATCC performs a really incredibly valuable task, but bless their hearts, they handle a huge number of strains, and getting a strain that is misannotated from them is not unheard of. Nor is getting a mycoplasma contaminated cell culture strain or a HeLa cell contaminated strain. It's a big problem starting in the 1960s. So I think what we've done actually is just found an error in ATCC's catalog that we can now correct, and sort of proves the point that I think this assay is working, or will be working soon. Limit of detection is extremely good. So we have some more replicates to do, but we're getting down to 2 or 3 genomes per reaction. And I think our validation plan is interesting. We've been able to bank some DNA where we've been able to identify incidentally Leishmania in the clinical lab. We'll also send some of our promastigote pellets through our extraction process. And then we have a number of FFPE cases where we either weren't able to identify a Leishmania species, a Leishmania organism, but it was identified at the CDC in that specimen or in another specimen for that patient. We still have some residual genomic DNA samples that we have extracted, and then there are 2 FFPE cases that I've seen recently where Leishmania cropped up in the differential, but I will be shocked if that's what it is, and so I want to know. So I want to move on to the last sort of vignette that I have in the last few minutes, which is what happens when I don't get a PCR result, there's fungus in tissue, and I want to try and make a diagnosis. And so I'll start again with this one case, which was shared by me with my colleague and teacher, Larry True. And for those of you who train with me, you might be wondering what on earth this is doing in a prostate. This was a cystoprostatectomy from a 62-year-old guy with a high-grade urothelial neoplasia. And there was one block of the prostate that was full of granulomas, including a number of these really well-preserved yeast forms, which in several cases appeared to have broad-based budding, a nice double-contoured cell wall, and they ranged in size from about 12 to 23 microns. And so, this sounds like blastomyces, but it's really hard to rule out Cryptococcus, particularly C. gattii, given that he doesn't have a travel history to a blastomyces endemic region. It's also hard to rule out definitively coccidiomycosis, although it is a bit small for that, and blastomyces is more likely to disseminate. At any rate, The patient did not have any lesions in their lungs and was not going to receive systemic chemotherapy. So despite the fact that we didn't get a PCR result, we wound up not pursuing species-specific PCRs for either crypto or cocci, which we thought about. And ID was sort of comfortable calling this probable blasto, but they recognized the pitfalls of trying to be specific, even with some of these sort of classic features. So thinking about this case and other related cases, where I don't know why we don't have a PCR amplification, I'd really like to develop this multiplexed fungal fish assay where we can encode on species-specific nucleic acid probes information using combinatorial fluorescent labeling. And so what I mean by that is basically if you mix up any combination of these 6 fluorophores, you can get 15 possible combinations, and so you can pretty rapidly expand the number of organisms that you can identify, or the number of targets you can throw at a sample all at once and image across multiple spectra. And so you could think about a couple of ways to deploy this, either as a single pool or as sort of multiple cascading pools of fish probes. This has been done in colonies, in bacterial colonies. So on the left, this is not my work, dispersed dental plaque, or semi-dispersed dental plaque. This is the real color image, or the fused image of imaging in 6 channels. And then after linear unmixing and sort of decoding these combinatorial labels, you can then assign taxons and get a false color image, and you can actually see where the different organisms are interacting. And so this is useful on the basic science side, trying to understand how complex communities interact. But I think for us, it really becomes a potentially powerful diagnostic tool when PCR doesn't work. So have folks done this? And the answer is yes. So first, in the clinical microbiology lab, we have FDA-cleared kits that we use either to detect mycobacteria, as we do here, or other labs will sometimes use these for detecting certain bacteria or endemic yeasts. Going into FFPE tissue is a bit more challenging, but folks have worked on at least genus-specific probes for the last 20 years. And high specificity is certainly achievable. I think we can do better. Work in Steve Salapante's lab, I think, is getting to that. And then, the sensitivity is also relatively high. Probably not as high as PCR, so not a replacement. But I think filling in a gap where sometimes all of your tools don't work. There are other ways to sort of biochemically enhance the binding, as well as to enhance the signal amplification. So the Pathology presents, there will be a fabulous speaker, Brian Bellevue, next week at 4:30, who's talking about saberfish, which is one of these sort of possibilities for sort of chaining together different molecules to encode more information and to amplify the fluorescent signal. So I will be there. And then I think the reason this hasn't taken a hold in anatomic pathology labs is, one, you have to see enough fungal disease for this to really be useful. And two, it's just, I think, been eclipsed by the excitement around PCR, followed by sequencing, and now next generation sequencing. Just a couple of very brief examples. So Kathy Montone's group at Penn developed both Aspergillus, Fusarium, and broad Dematiaceous probes. This is not fish, but ish, much like our EBV-ish probe that we use in hematopathology that many of you are probably familiar with. And so on H&E, you see that the Aspergillus and the Fusarium both look a lot alike. So having some sort of organism-agnostic test is really important. The Aspergillus-ish probe readily detects the Aspergillus hyphae, but not the Fusarium hyphae. There was a little background staining, I think probably just from the counter stain. And so she's published this in a number of ways over the last, decade. And then about a decade ago, our own David Frederick at the Fred Hutch worked with a European collaborator to compare fish in FFPE tissue to a broad-range fungal assay that they had in their lab that's similar to ours. And they found that the fish was worked, but was not as sensitive as a PCR. And there were about 3 of these 40 cases in which fish was positive and PCR was negative. And in all of these cases, the histopathology was positive. So I think there really is a niche for this assay. the way we've gone about trying to start this is with collaborating with my colleague Lori Barassa and the mycology staff, is to collect interesting, clinically relevant molds, cut them up, and embed them in as formalin-fixed paraffin-embedded tissue. And so you can see sort of the range of organisms, a bunch of different aspergilli in case we really want to get organism-specific and not just leave it at the genus level. Timor nephii and penicillium, Fusarium alternaria, and then a slew of, these are mostly dematiaceous molds, and I've used the pathologist trick of inking them so I can tell them apart and trace them back. And so I think this is a really nice library of tools that we can then use to develop this assay. I will also say it is really challenging to cut FFPE tissue. So for residents, be kind, cut your sections thin, and remember that this is hard, so appreciate your histopathologists. You may be able to see that these have been cut, and so we have slides, we have probes in-house, and we're getting going on that project. In terms of the use case, I think there are really sort of two opportunities. One is when PCR fails, as I mentioned. The other, you can imagine that this could be useful as sort of a perioperative type of rapid diagnostic. So the Montone group has been able to get the staining time down to 3 hours or less. So you'd still have to be able to cut them, but one could imagine having an intraoperative consult where frozen section is positive and then moving very quickly to this sort of fish assay. Again, patient selection would be important, but just trying to think ahead and decide when we might use that. So I want to close by just making sure I acknowledge people, and I apologize, I probably left people off, but I've highlighted Alex and Carissa and Kendall's involvement. Andrew Bryan was really instrumental in getting the fungal utilization PCR off the ground. Patrick and Nathan have been really essential in getting a lot of the informatics support for that. And then very helpful conversations along the way with Karen, Brad, Druba, and then Rod in AP, Ian and Greg in ENT. Druba and Kyoko have really just been essential sources of support both for me clinically as I get involved in the molecular micro lab, and Kyoko has been really essential in both of these projects, making sure we have DNA for validation studies, reagents to build our assays. And then I mentioned Ichi did sequencing. Steve and Brad have been involved in this project, and Steve on a little bit on the fungal fish project, more so in the future. And I want to recognize our collaborators, Fred, Wes, and folks in both their lab and in Steve's lab. And then finally on the fungal fish project, I've been working with Frank in Bazad's lab, and Bazad Najafian and Ram Akalesh, my collaborators in AP, have been kind enough to share their time and expertise with me. So I'm very grateful. I want to close by just acknowledging the molecular micro lab. This is a really fabulous team. I'm really lucky to be a part of this group. I think they turn out great results every day. And with that, I'll close and take questions. Thank you. Sure.
So in the first section, you said negative result is really useful, or in some people it's really useful, but you also said the size of the sample makes a huge contribution to the meaning of the data. So do you have anything more to say about negatives in tiny samples and negatives in big samples?
Yeah, so Sean is asking about sort of when do you determine whether a negative is a true negative and is therefore informative because you're ruling out infection, or it's just too small a sample and so you didn't get an amplification. I don't have a good answer now. I think, if you have pathology to look at, that's really informative. And so if it's a small sample, but there's no inflammation, or it's something that looks like, I don't know, lung rejection, for example, I think it's much easier to say that's a true negative. There have been a couple of cases where the first AFB stain that we got on a case had acid-fast organisms, but the pattern of inflammation didn't really fit. There's nothing in the PCR, and so then we went back and looked at it, ordered a follow-up AFB stain, which was dead negative, and so we really think that that was contamination of the stain. So I think trying to put all those together really matters a lot. One of the questions that I hope to look at is that precise question. We record the size of our specimens, and so you could also imagine that it goes the other way. So that prostate case that I showed, one of my hunches is it may have been negative because it was such a big sample that maybe we just gummed up the columns and it needed a manual extraction. So I think there are pitfalls in both ends of that spectrum, and hopefully the next time we're in this venue, I can answer that for you. It's one of my questions going forward. Thank you. Other questions? Susan.
That was a great talk, Josh. In your first part, you mentioned that you discarded a lot of the data because it wasn't sinus, and so I'm wondering if you have plans to go back to those data.
Yeah. Susan has asked whether the data that we didn't look at, non-sinus sites, is informative, and what are we going to do with that? And I think there are a number of really sort of immediate questions we can ask. I'm really interested in other pulmonary sites, right? And thinking about, if you have an invasive fungal infection, excuse me, or just fungus detected in your paranasal sinuses, is it in your lungs too? And there is some literature around that. I think we can ask questions about, for the Aspergillus PCR, what's the utility in that? Because it is a very fast turnaround time. It's used a lot in BAL and in pleural fluid and on lung specimens. So I think that's another really important question to ask. I think there's a wealth of mycobacterial data which was not included in that data pool but could be subjected to the same sort of analysis. There's a lot to mine in there. I'm sorry I don't have more information to tell you about it now. Yes.
Yeah, it sounds like there's a perception that it's difficult to get robust amplification from FFPE preserved tissue, but you found pretty concordant results between fresh and FFPE. Did you have to do any kind of special optimizations of the primer design, or do you think straightforward?
That's a great question. So do we do anything different molecularly for FFPE tissue versus fresh tissue? The short answer is no. I think the longer answer is we intentionally target, sort of try and minimize the amplicon length, because if you think about DNA shearing that's wrapped around histone shearing, you sort of get a size limit, and so shorter amplicons are better. But same primer set. We try really hard to get tissue for us to be the ones to cut the tissue, just because it gets handled a lot in a non-sterile way, and so we try and get rid of contaminating microbial DNA. But other than that, no, there's not a lot that's special that we do. Good management, good staff, and longitudinal investment in people, but maybe that's self-evident.
I have one more question. Yeah. So you had this 80% mortality when the treatment was delayed, like till 12 days. So it seems like there's a race for both sensitivity and speed. You're down to 5 days. I mean, are these people just presenting so sick that most of the pathology is a foregone conclusion and you can't get below 5%, 10% mortality? Or like if we had a 1 day result, can you estimate the magnitude of the clinical impact?
I can try. So the question is basically, at what point is fast enough for particularly diagnosing mucormycoses? And are the patients so sick that they're sort of already committed to a terminal fate? So I guess borrow from cell developmental biology. I think probably early on, there probably is real benefit to starting earlier, and it's not just this binary cut point at 6 days. This is only 70 patients. So it'd be interesting to see this with a much larger sample set. I think one of the problems is actually patients don't necessarily present that sick. And so if you have a little bit of eye pain and kind of a stuffy nose, most of us, I think, might ignore that as a seasonal allergy. But if you are 3 weeks out from chemotherapy, that's potentially a devastating symptom, even without a fever. So I think speed probably does matter, and getting down to 1 day or 2 day and starting therapy early matters. I think at this point, usually when people are highly suspicious for an infection like this, you get upfront empiric antifungals, often combination therapy, and then the question becomes, how quickly can we pull off the amphotericin to try and protect someone's kidneys, or how quickly can we pull off the voriconazole because they're having some sort of CNS toxicity? So thank you. That's a great question. All right. Well, thank you very much.